My name is Maeve Secor, and I'm a rising junior at Occidental College. I'm an undergraduate researcher at the Moore Lab of Zoology, and today I'm presenting my URC summer research project. The title of my presentation is How's the Weather Up There? Elevation-Driven Niche Divergence in a Bird Ranging from Mexico to Panama. A long-standing question of evolutionary biology is whether ecology tends to drive diversification. This refers to how the characteristics of an organism's niche influence how it selectively adapts to its environment, and how natural selection eventually leads to reproductive isolation and evolutionary change. The most famous evidence of niche divergence is exemplified by Darwin's finches. Darwin found that the original colonizing finch species spread into different open niches on the Galapagos Islands, and that then natural selection led to an array of beak shapes that helped the birds exploit different resources. The theory of niche divergence describes when the characteristics of an organism's niche influence natural selection, which leads to the reproductive isolation of a species. In contrast, the theory of niche conservatism suggests that speciation is actually more influenced by biogeographic barriers than it is by ecological differences. One of the most well-known studies of niche conservatism, conducted by Peterson in 1999, found that allopatric sister taxa, such as the blue mockingbird and the blue and white mockingbird, were separated by a barrier, the Isthmus of Tehuantepec in southern Mexico, and that they were occupying very similar niches despite their geographic isolation. For my project, I decided to focus on Fucatus, a genus of 6 woodland species with large bills that are mostly seed-eating. The first species I chose to investigate is the yellow grosbeak, Futicus chrysopeplis. Yellow grosbeaks are a species that show biogeographic variation in the subgroups of the species. Chrysopeplis is a prime species for a study of niche variation because it has multiple distinct geographic groups, and people notice them occupying a range of different habitats. I also decided to include the black-thighed grosbeak in this study because Futicus chrysopeplis is what we call paraphyletic, meaning all the members of the group don't share a recent common ancestor unless you include another species, Futicus tibialis. Tibialis is found down in Costa Rica and Panama, known as birding hotspots, which means that this species has a bountiful number of observations to help us construct its range limits. So here's a map showing you the ranges of the yellow grosbeak and the black-thighed grosbeak. As you can see, yellow grosbeaks are found in Mexico and Guatemala, while black-thighed grosbeaks are found down in Costa Rica and into Panama. Within the range of yellow grosbeaks, the break between two of its subgroups, Futicus chrysopeplis and Futicus chrysopeplis aurantiacus, is across the same isthmus of Tehuantepec, the biogeographic barrier used in the classic Peterson study, making yellow grosbeak's range a good system for testing the theory of niche conservatism. Advances in satellite technology in recent years have catalyzed the infusion of speciation research with large-scale ecological data collected by satellites. In 2005, the WorldClim database was established, giving us access to 19 bioclimatic variables that describe surface means of temperature and precipitation, seasonality, and potentially biologically limiting extremes. WorldClim data is collected from weather stations and then interpolated for the grid cells in between, giving us ecological data for every square kilometer of the Earth. With access to these data sources, we are now able to do large-scale ecology from space. Remote sensing satellites work by capturing photos and through infrared and radar technology to collect global data on the environment. This is incredibly important given the travel restrictions as a result of COVID-19, in addition to the fact that traveling to these places to collect climate and habitat data would be a major endeavor. So my first step was pulling the locality data on Futicus chrysopeplis and Futicus tibialis from the online ornithology database eBird, a publicly available source of bird locality information that allows users to upload GPS points for bird sightings. I started out with approximately 9,000 eBird observations, so I used the programming language R to thin my dataset. First, I filtered out all historical and incidental observations, which tend to be less accurate, and then I implemented a 5-kilometer effort distance filter and a 200-minute duration minutes filter. I cropped all points to a map of Mesoamerica to remove any points on islands. And lastly, I excluded occurrence points within 1 kilometer of an existing point, bringing the number of Chrysopepalus and tibialis observations down to a total of 352 occurrences. Then, I used R to extract environmental data from remote sensing satellites for each occurrence point. Ecological data included 6 vegetation variables and 8 climate variables at 1 kilometer resolution. 11 of the 19 world climate variables were removed due to high correlations with other variables. Once my occurrence data was thinned and I had extracted the environmental data for each occurrence point, it was time to begin statistical analysis. I used the software Stata Intercooled version 16.1. My objective was to compare ecological niches among the 6 groups to test for ecological divergence. I began by running univariate statistical analysis of each variable independently, and then I performed a multivariate analysis of all variables together in the form of a principal components analysis. Principal components analysis is a multivariate method that projects data in a higher dimensional space into a lower dimensional space by maximizing the variance of each dimension. So I want to start by showing you some of the single variable differences so you can get a feel for them. So here's canopy height across geographic groups. And here you can see that tibialis is clearly in a much taller forest than Chrysopepalus. We can also notice how group 3 is in an area with significantly lower canopy height than group 4, which is interesting when you consider how these two groups are allopatric sister taxa occupying niches on either side of the Isthmus of Tehuantepec, the same geographic area of the Peterson study, which found no niche divergence in this region. The 5s and the 6s are showing much higher canopy heights. And within this species, there also appears to be a greater canopy range in the heights of the western cluster of tibialis, group 5. So here we have annual mean temperature across geographic groups. And this is a very interesting pattern because the groups are seemingly resistant to the natural temperature gradient in which the mean temperature would normally increase as you approach the equator. However, here we have mean temperature decreasing as we decrease latitudinally. And at least partly, I think it's because the birds to the south are at higher elevations, which we can tell by looking at elevation across geographic groups. In elevation, the 2 northern groups of Chrysopepalus are at a much lower elevation. And it also seems that there's been some sort of niche shift from high to low, or vice versa. I also want to point out that the western cluster of Chrysopepalus group 2 has a significantly lower elevation than its neighboring cluster group 3, which occupies a niche at around twice the elevation of group 2. Additionally, the western group of tibialis, group 5, has a higher range in elevation than its eastern counterpart, group 6. So we're already seeing some kind of similar trends in different variables across geographic groups. Here we have annual precipitation across geographic groups. And this is another case where there's a lot of variation within Chrysopepalus. Group 1 and 3 of Chrysopepalus have much lower rainfall, and again, you can see that there's a noticeable difference between groups 2 and 3, the same subgroup of Chrysopepalus, with the 3s occupying a niche with lower precipitation, and in addition to the fact that they also are at a higher elevation with lower canopy height. And those 3 variables kind of make sense when you consider them side by side. We can also see that the western cluster of tibialis is occupying a region with higher rainfall than the eastern cluster. But you can also sort of tell from this that tibialis is quite different from Chrysopepalus. So when I went ahead to do the PCA, I decided I wanted to tease apart these kind of interesting but conflicting differences among groups 1 and 4 and leave tibialis out of the principal components analysis. So here's that PCA I've been talking about. And what we're looking at here, on the y-axis, we have PC1, which is characterized by a trade-off of heat and rain, with lots of trees and tall trees where the rain is higher and the average temperature is lower. And then on PC2, which is the x-axis, we have warm rains and seasonality in opposite directions, meaning one direction on the axis is places with warm rains and the other is places with more seasonal temperatures. But kind of when we look at it all together, we're seeing divergence in multidimensional niche space. The point here is that, of course, there is some overlap but the niches within Chrysopeplas are quite divergent. You can see that the 1s are occupying a totally different niche from the 2s, despite being neighbors on a map, and that 2s and 3s have a little bit of overlap, but are largely quite different. And the 4s and the 3s also have quite a difference in the space that they're occupying. We can also make note that the 1s are occupying a very compact niche space in comparison to the 2s and 4s, who are occupying a much broader niche space. This could possibly mean that the 1s are niche specialists, while the 2s and 4s are more niche generalists. But I think what's really interesting here is that between groups 2 and 3, which are taxonomically classified as the same subgroup, there appears to be quite a difference in the 2 niches of the clusters within that subgroup. So lastly, I have my conclusions. It seems that the grosbeak species have diversified in their niches across Mesoamerica. The key study on niche conservatism, conducted by Peterson in 1999, found no evidence of divergence across similar areas. If you recall, Peterson was also looking at the Isthmus of Tehuantepec, and the 2 species he found on opposite sides, he found their niches to be completely conserved. And that's very contradictory to the results that I got. It also seems that Fucatus has shifted their niches either up or down elevation, but we cannot know which direction without considering the evolution of the genus as a whole. Further study will be needed of the morphology and genetics of this species to investigate the consequences of niche divergence for the organism. This could mean looking at their beaks or their wingspans, and looking at their DNA to see how the characteristics of their divergent niches might be influencing their evolution. So I'd like to thank my professor John McCormick for being my mentor this summer, and I'd like to thank Ryan Terrell for his expert advice on computer science, and I'd also like to thank Professor James Maley for introducing me to the Moore Lab and for his help with my initial SRP proposal. This project was funded by the URC and Professor McCormick's NSF Career Grant. And lastly, if you have any questions, please send me an email at msecor@oxy.edu, and I will get back to you as soon as possible.